In this lesson, we're going to talk about somatic hypermutation, which is sort of a complicated and obscure sounding term, but in some ways it might be the most important part of the whole immune system. And maybe you're a huge fan of T cells or you just love neutrophils and you think I'm an idiot. But somatic hypermutation is how you make antibodies that can detect anything, and so I think it's incredibly important. And so to talk about somatic hypermutation, we're back at the training center, where archers hone their craft with precision and focus, and perfect their aim and technique. And that idea of honing and perfecting is what somatic hypermutation is all about. So to be a little more specific, somatic hypermutation is a process in which point mutations are randomly introduced into the B cell receptor in order to produce a hugely diverse array of receptors with different antigen binding capacities. And we've drawn in these hyped up B archers to represent somatic hypermutation. Now, do you see how they're circling back and forth between the dimly lit dark bunkhouse and the bright sunlit yard? That's because B cells travel back and forth between the dark and light zones of germinal centers in secondary lymphoid organs. They do that traveling back and forth once they've become activated. They undergo proliferation and somatic hypermutation in the dark zone, and then come out into the light to test out their new mutations. And like I said, this is all about honing and perfection. Good isn't good enough here. So B cells that can bind to antigen with good affinity keep cycling until a B cell receptor is created that has a strong and more specific affinity for the antigen. That's a process called affinity maturation. Now that we've established what somatic hypermutation and affinity maturation are, let's back up for a second. We'll go through this training regimen in more detail, and the first thing you should know is that somatic hypermutation can only occur in activated B cells. So here we've got dendrilox, which is our recurring symbol for the dendritic cell, communicating with some B archers. And she's there to remind you that, like helper T cells, dendritic cells activate B cells by displaying antigen with MHC and co-stimulatory molecules. Once a B cell is activated, it can go wild and undergo somatic hypermutation. And a key enzyme involved in somatic hypermutation is called activation-induced cytidine deaminase, or AID for short. We've added this AID, or Arrow Improvement and Development Station, here on the right to help you remember this super important enzyme. AID works by introducing point mutations, and specifically it turns cytosine into uracil in the variable regions of the B cell receptor's heavy and light chains. So, at the AID station, we have a young archer customizing their antibody-shaped arrow tips. Remember, the antibody-shaped arrows represent the B cell receptors, which are themselves antibodies, and the arrow tips represent the variable or antigen binding regions of those receptors. The result of introducing these point mutations is the creation of B cells whose receptors display a wide range of affinity for antigen, meaning some bind very strongly and some don't bind so strongly. And that means it's time for that receptor affinity testing that we mentioned earlier. The goal here is to select for B cells that have the strongest and most selective antigen binding receptors. So here on the left, we see an archer looking pretty sad because, well, his aim sucks. Unfortunately for him and all the rest of these amateur losers, they just don't stand a chance. Which means it's time for the pit of death. These B cells whose receptors have the weakest affinity to antigen are eliminated by apoptosis. Sucks to suck. Meanwhile, the B cells with the strongest attraction to the antigen will receive a signal to multiply. That's what you get when you hit the bullseye. This multiplication is called clonal expansion, and after multiple rounds of clonal expansion, we end up with a bunch of kick-ass B cells with peak antigen affinity and specificity. These B cells eventually differentiate into memory B cells and plasma cells. And all this sounds amazing, but hold on, because there's one more thing we haven't discussed yet, which is called isotype class switching. This is when B cells change the type of antibody they express. Class switching allows B cells to fight different types of infections or activate other immune defense strategies, such as complement activation. The key takeaway here is that AID is not only responsible for somatic hypermutation, but it's also important for isotype class switching. And so we've depicted that here. See the helper painting the long part of the arrow? That part of the arrow corresponds to the constant regions of the antibody. AID promotes class switching from IgM and IgD to A, G, and E. And just to help you further remember that isotype class switching is going on here, we'll put in our do some damage banner to help you remember those different antibody classes. Alright, enough training. Let's take a quick look back at what we've covered before we get out to the battlefield. Somatic hypermutation is a process that happens solely in activated B cells and involves the random introduction of point mutations in the B cell receptor, or antibody. After somatic hypermutation, receptor affinity testing is conducted to select for B cells whose receptors bind to the antigen the best. The end result is affinity maturation, that is, the creation of B cells with a stronger and more specific affinity for the antigen. The B cells with the strongest affinity for the antigen will undergo clonal expansion, multiply, and go on to differentiate into either plasma cells or memory B cells. And finally, don't forget that the enzyme AID plays a key role during somatic hypermutation and isotype class switching, which is when the constant region of the antibody is changed and the B cell goes from producing IgM and IgD to either IgA, IgG, or IgE. With all that high-intensity training and all those friggin' letters, I need to take 5, or maybe 10,000. Hey, does that aid station have any cots? Asking for a friend.